This is episode 310 of the Stem Cell Podcast, Epithelial Organoids with Dr. Hans Levers. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us. Leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have an icon of the organoid field, Hans Clevers. He's from the Hubrecht Institute, and he's here to talk about his research on intestinal organoid models, as well as, just pick a tissue. He's got an organoid. So we're going to talk about it all. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up in a second.
But first, registration and abstracts are open for the Global Stem Cell Event, the ISSCR 2026 annual meeting, 8th to 11th of July in Montreal, Canada. Join nearly 3,500 scientists, clinicians, and industry leaders for 4 days at the frontiers of stem cell science, regenerative medicine, and engineering biology. From fundamental discovery to clinical breakthroughs, this is where collaborations start and new ideas take shape. Submit your abstract by February 25th for a chance to present your work and be considered for travel and merit awards and design your dream session. Also, you can submit a science spotlight proposal by the 25th of January as well. Be part of the global conversation shaping the future of stem cell research. Visit isscr2026.org to learn more. And indeed, we are very excited about ISSCR 2026 in Canada A. We are going to be there for sure. And one thing that's also going to be there is organoids, of course. Organoids, brain organoids in particular, are going to be there well represented, I'm sure. Check out the poster sessions, no doubt. Check out the plenaries. You're going to see organoids all over the place. This is a Nature Neuroscience paper that I'm going to start off with in the roundup, believe it or not, coming from the lab of Tal Sharf over in my neck of the woods, just up the coast here in California, UC Santa Cruz, home of the banana slugs. You know that they're known as the banana slugs, Dalen? Well, now you know. Now you know their mascot is the banana slug. Also, of course, Santa Cruz is very well known for their internationally recognized genome browser. I'm sure you've used it. We've all used it. But they do great organoid work as well. Like I said, this is coming from the lab of Tal Sharp. First author here is Tise van der Molen. Apologies there. This is a cool story that I wanted to cover because it's, of course, about organoids, but also it's about early brain development and the earliest of the early when it comes to brain development, in particular brain functionality. So, neuronal firing sequences, the neurons fire. That's the foundation of how the brain works, certainly. And understandably so, we haven't really been able to figure out how that happens in the beginning, the very beginning in humans. Of course, you don't have great ways to uncover that until you have organoids. Of course, organoids is a unique way to study brain functionality, neuronal functionality, circuit connectivity, all these good things. So, what they're doing here is trying to figure out, using organoids, how these initial firing sequences happen in the brain. And yeah, so neural firing sequences are thought to be the building blocks of information and broadcasting in the brain. I mean, the analogy is to the computer. The computer is also dictated by electrical inputs. The brain has electrical inputs as well. But like I said, it's unclear when these sequences, these firing sequences, actually emerge during the development of the brain, very early development. And here they're actually showing that they're structured firing sequences that appear in the spontaneous activity of both human and mouse brain organoids. So there's structure to that initial firing. It's not just random. Apparently, there's some sort of pre-coded structure, guided by evolution or whatnot, that is emerging at those very initial firing stages in the brain. And this is true in both the unguided and the forebrain identity directed as well in terms of these organoids. And also, you see it ex vivo in neonatal mouse cortical slices. So in these ex vivo slices, you see the same thing. It's not just random firing that are those first firing signals, it's actually structured nodes of firing coordinated. And they show that there's temporally rigid and flexible firing patterns in both these human and mouse brain organoids, early postnatal murine somatosensory cortex, but not dissociated primary cortical structures. So you have to maintain that tissue architecture, it seems like, to have these proper firing sequences to happen. And so this is all suggesting that these temporal firing sequences don't arise in experience dependent manner. That's important here. It's not that early natal experience, prenatal experience that's dictating the sequence of these firing. It's kind of coded in already. So what they describe is they're constrained by what they call a pre-configured architecture, quote unquote, that is actually established over the course of very, very, very early brain development. And so they demonstrate this developmental recapitulation of the neuronal firing patterns. And ultimately, they're hoping to highlight the importance of brain organoids, of course, not just for studying adult disease, but in particular those early phases of neurodevelopment, and also as a model for circuit assembly. I mean, Sergio Pasca is working on this in some capacity when it comes to the assembly of circuits across different organoid types. So here we're talking about intra neural organoid circuit assembly and the level of the individual neurons themselves, and actually when they start kicking off. And the analogy here is, and of course I'm going to make the cardiac analogy, is in the cardiac organoids we all wonder a similar question, like how those initial contraction sequences in the organoids and in the heart, what are they dictated by? Is there some sort of pattern to the noise, or is there more of like a structured pre-organized situation like what they're showing here? So it's an example, and yeah, an example of how these organoids can be used to uncover that very early black box of human development, and that's why I like the story.
Yeah, I like this story too. When you sent it to me, I gotta be honest, initially I was like, grown. Now wait until the antis get a hold of this study and they're gonna use it as like evidence of there's circuitry, there's firing, neuronal firing, and like as a proxy for personhood. And I hate to bring it there. It's a science show, but I can't help myself because that's what jumped to me. It was like I remember when Mautry came out with that. The organoids, maybe not that they're thinking, but they're firing, and then everyone jumped on that and went kind of crazy. And I think scientists were a little bit like, "Hey, let's not assign any kind of personhood to these organoids. We got to be more careful." And so when I saw this, I was like, "Here we go again." But quite the contrary here. I think that, and the science is super strong. I think you did a great job of speaking to the impact and how this is really important to establish this as a precedent and as a model to build these kind of more practical, I would say, clinically applied approaches incorporating organoids, assembloids in terms of neural circuitry versus like when do neurons first fire, which maybe someone would think is like an academic question. So yeah, I don't want to say this is just academic. You did a good job of explaining the impact, but from like that ethical, philosophical standpoint, I think this even maybe makes the counterpoint here. Use the heart as a analogy, and I think the difference there maybe, and I didn't read this closely enough, so maybe I'm speaking a little bit out of turn in terms of what the authors might say. But like the heart, you can recapitulate that kind of cardiac contraction in the dish, and you can see it, and that's such a vivid illustration of that's why a lot of us fell in love with the stem cell system, pluripotent stem cell system in the first place. But here, I think one of the major points, my takeaway, is that these dissociated cultures, even if they were firing, you dissociate them and they lose that kind of capacity, which is not just like, okay, once assigned, it's they're endowed with it, even if you take them out of context. And I think that really underscores, as you said, the essential nature of that architecture. That is what makes up the thinking brain, is to extend that forward in development. So, like, I would use as an argument that we're not even close to the architecture of thinking or whatever, even pre-thinking brains in our organoid and model systems. So this whole conversation, and while we do need to be mindful, of course, and approach it carefully and have all the committees, etc. But like the idea that we're getting awareness, personhood, any of that stuff, come on. The architecture of biology and the matter to mind transition is a lot more complicated than we can assimilate in a dish. So I loved this story for that reason. I'm gonna put it in my pocket as an argument against this whole dramatic apotheosis extension of the thinking organoid. Like, come on.
Yeah, I mean, people are gonna spin it however they want, but I agree with you. I think we all know that it's always a stretch to make that comparison, to make that analogy, to quote brains in a dish, which they're not. They're far from that. And this is a, like you said, a data point that's further indicative of that. But still, even in these ultra simplified scalable models, you can learn so much about how circuitry forms. And that's the intent and purpose here.
Yeah, and that's a good point that although we're not talking about the complexity of mind, neural firing in itself and circuitry involved is beautiful and miraculous. So this is a little peek into that black box and sure to bear a lot of fruit and studies like it as well. I'm going into organoid for organoid, and of course, with our guest today, apropos. Although we're doing more pluripotent cell derived instead of adult, but we're gonna get into all that stuff in a few minutes. My story is a quick one in DevCell about vascular organoid models, which we've talked about forever since the days of Joe P. back in COVID, when he used these vascular organoids to model infection and that tropism for the vasculature. Here, this is expanding upon that, and of course, in the intervening years, these vascular organoid models have gotten more sophisticated, incorporated multiple associated cell types, parasites, even immune cells, to model different aspects of the vasculature. In this case, it's I don't want to say simple, but it's more straightforward, right? And it's really just about the it's a very endothelial centric analysis of disease and aging, but using progeria as a proxy for this accelerated aging, right? And what we're studying here is the aging vascular system, right? Because there's a lot of diseases that are attributed to vascular dysfunction and the structural functional deterioration that leads to these kind of microcirculatory defects, coronary heart disease, stroke, hypertension. I mean, those alone account for the majority of death in the developed world, but there's also, fundamental aging and its contribution to disease in many organs like kidney disease. Also, looking at like metabolic syndrome, there's definitely a component of that interface of the vasculature and the blood and sugars, all that. Uh, what we're modeling here is Hutchinson-Gilford progeria, which is practical in this case because it's this monogenic, devastating pediatric laminopathy that is driven by LMNA, laminin 1 mutation. And as we all know, results in this accelerated aging phenotype with premature, profound vasculopathy. And ultimately, that's what kills these kids around the age of 14 years on average, are these cardiovascular events. Um, Now, how do we study this? These patients are extremely rare, of course. There's limits on access to human tissue fundamentally, but the rarity of these patients means that we don't really have access to tissue, even postmortem or primary, readily. So, in this case, a broad group here from China, including and led by Jingshu Weishi Zhang and Guanhui Lu, And of course, on the list, we got Juan Carlos Izpizua Belmonte. I always have trouble with that. JC. Why do you have to have all the names, partner? Anyway, he dipped in here from Altos. Of course, if it's an aging thing, he's got to have a hand in it. But what they did was really straightforward. A short article on DevCell where they targeted the laminin to mutate with the same mutation in these Hutchinson-Gilford patients, and then they made these blood vessel organoids from these mutant embryonic stem cells, compared them to the wild type from which the cell line originated, and showed, of course, they could recapitulate the vascular defects, and mechanistically they went in and looked at this specific factor, serum response factor, uh, and showed that it regulated these angiogenesis-related genes. So, kind of drew the connection between the laminin loss and this mediator, serum response factor on the vasculature, and showed then that if you overexpressed this SRF, you could rescue some of that endothelial dysfunction. And finally, although this was in the middle of the paper, but I think in terms of impact, I think to confer the story with a little bit more clinical relevance, they showed that even in just naturally aged primates and outside of the context of disease, that this same kind of phenotypic serum response factor phenomenon is present. So this is, of course, this hyper extra model of vascular dysfunction, accelerated aging. But I think the idea here is that perhaps some of these same pathologies that are accelerated in the context of progeria may be at play in just normal garden variety aging vasculature. And here they provide this kind of therapeutic approach. While I'd say it's a stretch, this is more a model than a therapy. I think that maybe is the next tip, or the next, point on the string here is see if we can maybe, amp up the vasculature. And my kind of Trojan horse here with organoids and tissue engineering being bottlenecked so significantly by the lack of vasculature, I wonder if we can use these kind of insights to create a kind of super physiological endothelial cell. I mean, scary when you think about the evasiveness and potential for pathologies associated with that. But at least in the context of in vitro organ engineering, I like the idea of amping up endothelial cells to confer some little bit of extra function on them. So a lot here, some of which I kind of shoehorned in, but I think for a short article, it is pretty juicy. Arun, what's your take?
Yeah, cool stuff. I think progeria has always been viewed as such a unique disease because you're fast forwarding the clock of aging, right? Which, of course, is something that we hope to reverse. So I think there's a lot we can learn from the mechanisms of this disease. And I know a lot of people have been doing, including Juan Carlos, have been honing in on progeria as almost like as an example of what to target in a way, and how to, if you're trying to target aging, you got to target the reverse of aging, accelerated aging, right? So the mechanisms are sort of interesting to unravel in that way, but I mean, the big part of why I like this study is the utility of the model, and I think you're alluding to that. The utility of the vascular organoid model, and we're using this in our lab for other vascular disease modeling approaches, not for progeria, but for other vasculopathies, but it's becoming such a hot topic, these vasculopathies, and in particular these vascular organoid models, because they're not just powerful in themselves as models for disease, but you can do things like fuse them, right? We talk about papers using vascular organoids all the time to create organoids with enhanced vasculature. So they're powerful systems in addition to their inherent utility. It's their utility across different organoid types. I think the SRF overexpression and how that rescues the vascular I think that I agree with you. That's a bit of a stretch, but cool concept nonetheless. But I like the study because of the organoids. I'm sorry.
Yes, I would agree with you, and I'm glad you mentioned that aspect of these vascular organoids being incorporated into all these diversity as an accessory cell type because that again, as I kind of was getting at, I think is going to be the ultimate application of endothelial cells in tissue engineering is like what they do all by themselves. I think for a cell that has such amazing diversity and capacity for self-assembly and like invasion maybe in a positive way, being able to integrate and assemble an existing tissues through these like neoangiogenic processes like I think that maybe we haven't done enough with endothelial cells, but that's coming from someone who's tried many things and failed with EC. So really high ceiling for these cells, but I think there's a lot of fill-in that we need to do before we can really take them to their ultimate use.
Yeah, they're more than just plumbing, boss, as Shaheen
That's
has
correct.
always said.
You
But
said it,
sadly,
not me.
but sadly, I will say I think most of us just use them as plumbing. Sorry, Shaheen. I hope you're not listening to this. Moving on
He's not.
to he's not. He's not listening. We're good. Maybe he will one day. I don't know. Moving on to another favorite topic of mine, not just organoids or vascular organoids, but space. Of course, the
Space.
final frontier, space. 250 miles up. It's not that far. It's just in the z-axis, right? We're going up, not XY. And that's how far up the space station is. It's only 250 miles floating above us. And but of course, it's such an incredibly unique environment in that we're able to experience microgravity. And when you take gravity out of the equation, so much changes when it comes to cell biology, human physiology, and astronauts, of course, have been going to space for decades now, and we know what happens to their bodies. Not good things. A lot of bone muscle degradation and immune dysfunction too, which NASA has documented for many years. But you know, we haven't gotten to the nitty-gritty that we're talking about mechanism, right? We haven't gotten to the cellular mechanisms as to what actually happens to astronauts in space. And this is what our friend of the show, Kat Jameson, and her team down at UCSD, Sanford Stem Cell Institute, this is what they're hoping to unpack. And you guys know if you've listened to the show, like this is a lot of what I do in my day job here at Cedars-Sinai as the director of our new Center for Space Medicine Research. Is we do similar stuff. We try to see what happens to cells in space, how you can harness microgravity to make things in space that you may not be able to on the ground. And Kat's a dear colleague of ours and is approaching this in different ways, but is also doing phenomenal work. So that she's actually published a number of high profile papers on this topic as of recently. She actually just had another cell stem cell paper in similar space related research. But this is a study that I've seen over the last 5 years kind of come together over multiple missions to the International Space Station. And it's really cool to finally see this in print in Cell Stem Cell. Title here is Space Associated Stem Cell Hallmarks of Aging and Resilience in Astronauts. So pretty big title there. The first author is someone who I know pretty well, Jessica Pham, who actually was at a conference with me just 2 weeks ago. And like I said, last author here is Kat Jameson. So, like I said, people have known what's happening in space, whether it's immune dysfunction, cytokine dysregulation, telomere alterations, potentially, as was the case with the twin study from the Kelly brothers, the astronaut Kelly brothers, who are twins. So there's a lot of what happens in space, but we don't know exactly what happens to the hematopoietic stem cells, right, which are really critical to driving some of these immune phenotypes, of course. So they're looking at that. They were looking at the stress of space and microgravity on HSPCs and how resilient they are and how they're able to potentially maintain hematopoiesis and immunity over the course of space flight, right? So they did something relatively straightforward, but nothing is straightforward in space biology research. So they performed an HS hematopoietic stem cell functional characterization pre, during, and post flight from different samples that were taken during each of those time points. Right, multi-omics analyses of aging and resilience is what they call it in 9 astronauts. And again, N of 9 is not a huge sample size. We acknowledge that, but it's hard to get astronaut samples, period. And take that from me as somebody in this field. N of 9 across three different space flights is a huge accomplishment. I think part of the reason that this is in cell stem cell is most missions and most research projects to the ISS are an N of one, N of one or N of two, which is of course not how you want to do things for the sake of science. But they're able to get samples from nine different astronauts over the course of three different missions that were conducted with a company called Axiom. Axiom Space is aiming to develop one of the first private space stations out there and actually have sent private astronauts to the International Space Station. So these samples were taken from those nine private, quote, astronauts, not NASA astronauts, right? So what do? They took blood samples from these astronauts, like I said before, during after flight, did whole genome sequencing, telomere length analysis, mitochondrial profiling, transcriptomic sequencing, all the omics, single cell, cytokine arrays, um, and we're able to assess HSPC immune subpopulation survival dynamics. And they ultimately showed that spaceflight is associated with some reversible changes in hematopoietic stem cell survival, self renewal. As you might expect, it's a stressful situation. So there would be changes, of course. But a lot of these changes, interestingly, revert back to normal when the cells came back to the planet, which again kind of makes sense with seeing what happens to the astronauts at the macro level. So they saw changes in particular with this 8R1 gene, telomere maintenance, mobilization, cell cycle, a quote fight or flight profile of gene expression, and then they also showed some from some chip actually, clonal hematopoiesis, ApoBec3 activation, retrotransposon deregulation. So it's a broad omics level study of what happens to HSCs in space, and I think in my mind the big takeaway is that yes, there are stress and space-induced changes in HSC populations, but for the most part, these changes revert back to normal when astronauts come back. But the bigger question here is that what happens during well, I think two big questions. One, what happens during longer missions? These are really pretty short missions that these private astronauts went on to the space station just for like, 10 to 20 days, I believe. We were actually a part of one of these missions, the Axiom 2 mission, which is only for about 10 days. Um, and two, before I get to two, NASA's hoping, of course, to send astronauts to the moon, and all those are going to be much, much longer missions. So you got to kind of get that extended time point because this is almost like a transient phenotype. And two, just on that exact same topic, when you go to the moon, when you go to Mars, you not just have microgravity, lack of gravity in space, but you have a whole hell of a lot more radiation. And radiation is going to cause, I think, more severe changes in these stem cell populations. And it's kind of scary. No one has done these studies, and we're going to be sending astronauts to deep space and very soon in the Artemis 2 and 3 missions. And there are some in vitro experiments that are part of those missions to the moon and to kind of get to the bottom of what happens in response to radiation on bone marrow populations and stuff like that. But I think that's the bigger question here that, this paper is starting to get to, but we're not going to know. We're not going to know until those astronauts go, stay, and come back as to what happens to their HSCs and different stem cell populations and their radiation responses. And, certainly astronauts have gone to the moon already, but we haven't done any of the in-depth characterization of their cells. Of course, that was back in the 60s and 70s. So I guess stay tuned. I'm sure Kat and her team are going to be thinking of a part two for this, for the moon missions as well.
Part two, true. Call it a part three. I mean, the
Yeah.
the story that came out preceding this, we covered actually in the roundup, episode 304, which was just a couple months ago. So, yeah, I mean, these studies are so long in the planning and logistics, and a lot of kind of delays and disappointments, a lot of hope, uh, and it's a heavy lift, pun intended, but ultimately, I think the return is there, right? Not just in terms of scientific productivity, as evidenced by Cat's output over the last I don't know, couple of months, with more to come for sure. But also, I think I want to emphasize this point to your kind of let's see about the later, longer, and staying all that. Yeah, we're in space. I'm really going with the puns today. But the impact here to me, it's not just the data. And by the way, like n equals 9 is great for this study, considering how much you need to do, but it's limiting. But also, I love the before, during, and after, right? These kind of internally controlled uh, time points and during space flight, which gets to my second point, which is that the capabilities, the will to do these kinds of experiments in space, I think is very different from, the first foray, many forays into space, into the moon, where it was just like, "Let's go." Right? And, you look at the averages, astronauts live on average longer than most people, but of course, their fitness is tremendous. So that's not in any way an indication of what the impact on their health and longevity is. I think there's still that a lot of that remains to be seen. But here, the capability, the will, the proof of principle in terms of results, um, and not for nothing, but like that transitory element of it, I think is really key in terms of just, peace of mind, establishing at least an upper limit as we know it right now for is 20 days, if that's what it is. 20 days, you get reversible uh, phenotypes in terms of hematopoietic aging. Like that's great. Now we know that. So I love how much went into this, and I'm very bullish on how this kind of moves the goalposts on where we look moving forward with these space-based studies. I think the sky's the limit, or I guess I should say we're going even beyond.
The sky is not the limit. That's the analogy that you should be making here, which trust me, you think of a space pun, I've made it. So it's not news to me. But yeah, you're totally right. Like back in the day, we just wanted to go there just to say that we could, just to say we could be the first to go to space, the first to go to the moon, and so on. And we did care. Of course, we cared about what happened to the astronauts, but we didn't take a deep dive into their physiology and definitely not into their cell biology because we didn't have the resources and the analysis capabilities that we do now. And so now with the NASA Artemis 2 and Artemis 3 missions that are coming up probably in the next year, we're going to be going back to stay and we're going to have all of these amazing biomedical research capabilities at our fingertips to really take a deeper dive to what happens to cells in space. And I'm just so excited to play a part in it, and Kat's definitely leading the charge too.
And I know secretly when you say we, you're thinking you. You're thinking me up in Axios chilling. And I
Uh,
hope that that comes, man, because you deserve it, and you'd be great in space. You're the perfect fit, literally.
well, thanks, man. I don't take up that much mass. That's the other advantage. I'm not very big.
More puns, the perfect fit. Moving on, this idea of reversibility, of course, that's key in terms of peace of mind. But what it tells you is that these phenotypes can be reversed maybe intrinsically, or perhaps you can enforce that reversion, resolution, rejuvenation. And maybe that's another target for these astronauts or anyone who wants to be spacefaring, Arun. Wink, wink, wink. And of course, the best proxy for this, space aging hematopoiesis is aging on Earth. And it's associated, of course, with this decline, not just in the hematopoietic system, but all the tissues, a decline in stem cell regenerative capacity. And of course, that underlies the reduced tissue integrity and functional potential in specialized tissues. Um, and not just that, but you get these kind of aberrant growth, cancer, and other chronic disorders like CHIP, leading to imbalances in the hematopoietic complement, like this myeloid bias, et cetera. Even can lead to like cardiovascular disease. So the implications of the aging hematopoietic system are profound, and what causes the defects are myriad, right? There's intrinsic as well as extrinsic factors. But bottom line, in terms of what it results in, unlike young uh, hematopoietic stem cells, which are mostly quiescent. And therefore, that quiescence confers on them this higher ability to reconstitute the blood, kind of counterintuitively. But with age, that quiescence is lost in a lot of the hematopoietic stem cell, not all of them, but a lot of the hematopoietic stem cells. And of course, that's associated because they're growing with increased load of somatic mutations. You also get CHIP, this clonal dominance that favors myeloid populations, and a lot of other sequelae that are major malignancies, not just cancer, all kinds of hematologic malignancies. Um, so yeah, I mean, the intrinsic defects are myriad: epigenetic deregulation, telomere shortening, oxidative stress, genome and instability. You got mitochondrial dysfunction, metabolic dysfunction, alterations in defective autophagy. I mean, it goes on and on and on, and that's just intrinsic. Then you go to the niche. You got all the different niches, of course, need to be addressed, and you've got the stress, inflammation in those niches, and dysfunction in the constitution of the supporting cells with age, of course. So yeah, there's all these different things that converge on this reduced quiescence, and there's been many approaches targeting one or multiple of those deleterious influences, and they've had some success in restoring the youthful phenotype to hematopoietic stem cells. But and this is important, like, while they can confer, like, a phenotypic advantage, there's no intervention that restores the hematopoietic stem cells' potential for self-renewal, while also, increasing their regenerative capacity, right? So it's kinda like, again, counterintuitive or a little bit paradoxical that you need to make them self-renew more, but you also need to make them more quiescent so that they can reconstitute better. It seems like, you know, two opposing ideas even, right? So here we go. This is a story. It's in Cell Stem Cell, and it's based on lysosomes, right? So lysosomes, which are the centers for degradation and clearance of damaged or old macromolecules, organelles, they also have this. They're a storage facility, like a depot, but they also form these signaling hubs, right? Particularly for mTOR signaling, which is a key regulator of cellular metabolism. And not surprisingly, then perhaps there's mutations in lysosomal enzymes that result in these lysosomal storage disorders, which are what underlie a lot of neurodegenerative and metabolic diseases, right? And there's evidence that's been emerging that lysosomes are really crucial for regulating cellular quiescence, right? And this is where the group comes in. So, Sagi Ghaffari, excuse me if I mispronounce her name, who's from Icon here in New York, Mount Sinai. Uh, the group recently found that lysosomal degradation process it's linked, fundamentally linked to the potency of hematopoietic stem cells. But that link being, there's this correlation. The causality remains outstanding. What the contribution of the lysosomes are, apart from their just passive mediation of autophagy, it has not been defined. So here, Ghaffari's group teamed up with Mikhail Meniger, who's at INSERM in France, Paris, and they delved a little bit deeper into this lysosomal connection, showed that specifically lysosomes in aged hematopoietic stem cells are hyperacidic, they're depleted, damaged, and activated in this aberrant way. Um, and more importantly, that if you suppress these hyperactivated lysosomes using this small molecule drug, this vacuolar ATPase inhibitor, you can restore this kind of more native homeostasis in old hematopoietic stem cells. Mechanistically, they show this works by improving lysosomal processing of mitochondrial DNA in attenuating this inflammatory signaling that goes on. And here's the key. they do this ex vivo expansion using this inhibitor, and they get old hematopoietic stem cells, and they boost their in vivo repopulation capacity by eightfold, right? And now that, I mean, the numbers are like modest, but this is unprecedented. There's been a lot of small molecules that do this, and ultimately, the hematopoietic reconstitution and number of like stem cells in the pool are key metrics. But here, I think there's a lot of due diligence in showing that it was based in quiescence. They really went to cell cycle, measuring the number of cells that underwent 0 versus 1 or 2 or more um, cytokinesis events, and showed that this inhibition was able to choke out, or I shouldn't say that, sounds bad. They're able to instill like a much higher degree of quiescence. So it's not just the endpoint here. They grow these things for 4 days in vitro and then transplant them back at the eightfold reconstitution. But the mechanism and how they show how it works, and also this kind of out of left field idea of lysosomal regulation as I mean, maybe as a clinical inroad for this holy grail of ex vivo expansion of hematopoietic stem cells. Like, I don't think a lot of people would have predicted it. So I love the novelty. I love the clinical impact. This story, I tell a hematopoietic story pretty much every episode, Arun. They're all the best, but this one definitely the top 5% of even those.
But would it still get funded, even if it's top 5%? Oh, oh,
Ooh, not
oh,
not this year.
not this year, man. I also like the story because, yeah, it's kind of a nice connection to our prior story and prior stories when it comes to HSC aging and aging processes in general. And, to make a very bad analogy here, when the trash doesn't get picked up in Brooklyn, in your neck of the woods,
Oof.
people don't get very happy, right? The cell that is Brooklyn is definitely very upset. And that's the analogy here. When the trash gets messed up for the HSCs, when there's that lysosomal dysfunction, because it is the trash can of the cell, quote unquote. It does more than that, but certainly it plays a role in regulating proper agency activity. And I like this concept of the ATPase inhibition, but they say it themselves. They still need to take a deeper dive into the mechanisms there. They don't really figure out how those mechanisms are altered when it comes to lysosomal hyperacidification, hyperactivation. Anytime you have an inhibitor like that, it's going to do a number of different things that you got to dissect. So I think they're going to stay busy in that way.
For sure, for sure. But again, I mean, another strong point that you just made there, elevating the lysosome beyond just the trash. It's not just trash can, boss. It's not just trash can.
Love it.
Anyway, yes, it remains to be seen, the clinical impact, and that's where I always get tripped up, Arun. I'm just counting my money already. We're curing diseases all over the world. But then we haven't done a single large animal trial now, have we? So we'll see how that works. Um, Plenty to talk about with Doc. Probably nothing that we talked about just in the roundup, but I mean, we talked to Dr. Clever Hans for hours if he'd let us. He's got a pretty tight schedule, so we're going to get to it in just a sec. First, quick message from STEMCELL Technologies. Are you looking to add more physiological relevance to your research? Intesticult Media provides a complete workflow for establishing, maintaining, and differentiating human intestinal organoids. Use intesticult organoid growth medium to establish and maintain organoids in a more proliferative state. Then, to achieve more physiologically relevant proportions of differentiated cell types for your experiments, passage your cultures in intesticult organoid differentiation medium. Receive an offer to try Intesticult in your lab by visiting us online at www.stemcell.com/try-intesticult. All right, you guys, with us today, we have a legend in his own time, Dr. Hans Clevers, who is professor of molecular genetics at the Hubrecht Institute. His group studies the biology of Wnt signaling in tissue turnover and in cancer. The discovery of LGR5 as a generic marker of Wnt dependent stem cells within multiple adult tissues by his group has led to the development of technology to grow these stem cells into ever expanding epithelial organoids. Patient-derived organoids hold promise to predict drug response in a personalized fashion, and in combination with genome and editing technology, these organoids open up new avenues for the utilization of autologous adult stem cells to cure genetic and degenerative disease. Dr. Clevers, thank you so much for joining us on the show today.
Thank you.
Yeah, thanks so much for being here. Again, it's been probably more than 100 something episodes since you've been here last, but who's counting? Like Dalen mentioned, your lab has really fundamentally transformed how we model human tissues by creating organoids from adult stem cells. And everybody around the world is starting to use organoids these days, my lab included as well. So, you've done so much, but we're really interested in what you're working on right now. So could you start with there, summarize what your group is focusing on right now?
Yeah, so to be honest, I'm actually just in the process of transiting back from Switzerland. The past 3.5 years, I've been the head of what's called PRED, Pharma Research Early Development at Roche, where we, by the way, have also set up Matthias Lutolf as the first founding director at the Institute of Human Biology, fully focused around in vitro human models, so organoids, organelle chips. But so I've kept an eye from a distance on my lab in Utrecht. I stepped down from the executive role that I had in Roche end of August. So we'll be there still till the end of the year, but slowly growing back into my own lab here. And I've led it from a distance. So actually, they haven't stopped publishing in my absence. They never fail to point this out to me. So, but I do think I still contribute to what they're doing in the lab. So yeah. So what do we do? So the model has been. Gut, as you mentioned, I think in introduction. So my lab found one of the components of the Wnt pathway. This took us to eventually realize that Wnt drives stem cells. This is a long time ago. Also drives cancer in the same organ, colon. Then we realized that from what was known about the stem cells there, they're quite incredible. They are extremely active, far active than probably than any other stem cell in the human body. And this then prompted Toshisato as a postdoc in my lab to see if he could grow them. And then there were many surprises. One of them is that actually they don't just multiply, but they make little organs. They make these organoids. So when that worked, it took us a while to realize that it was not unique to the gut, but actually we could do this in different organs. And I think what has been the model is extremely pragmatic or maybe opportunistic. Basically, a new postdoc or a new PhD student thinks of a new tissue that we haven't yet looked at. Then we find a link to a clinic that basically could give us access to patient material, maybe has an interesting question to ask if we can turn that into an organoid. And then basically that person will then optimize culture conditions, typically takes half a year or so, to a state where we can grow them at least for half a year or something like that, where we can CRISPR them. Hopefully we can clone them. That helps a lot. And then we come up with some opportunistic question that where people have a strong belief one way, and we basically think, well, maybe it's slightly different. And then we test this in organoids, and then we publish a paper. Typically, we find a collaborator in that field, because every field I learned has its own culture, its own rules of how to write down things, what to cite, what the control experiment should look like, et cetera, et cetera. So typically, if you have a good collaborator that is anchored in a new field, we work together. We bring the technology. They often bring the sort of the real in-depth knowledge of that field that would be new to us. So that's been the model. And currently, there's somebody to give you maybe go through what I've heard here. Somebody is growing snake lung. We were hoping to grow snake gut because snakes, these constrictors, they can actually eat a pig, and then once they finished eating the pig, the gut gets very huge and they digest the pig, and then it doesn't need to eat for half a year or so, and then the gut basically shrinks again. So they were hoping to study that process, but now we have snake lung growing. And so we diverted this now into a nidovirus program, which is a very nasty virus, which there never was a real good in vitro model. And these snake lung organoids basically can be infected with that virus. But I think this describes a little bit how we work.
Yeah, that's a great snapshot, both at the overview level. I love that narrative synthesis, which vastly, I think, oversimplifies, but appropriately in a frank way that I love about the way you describe your science. Just the pragmatic approach and opportunistic questions that arise, but all the way to the granular level, the snapshot there with the snake lung. I mean, what a great synthesis. And we've talked about them all. I'll tell you, we've talked about all the organoids, not all of them, but many of the organoids. You can't talk about them all. We talked them all on the show. We've also talked on the show a lot recently about what a remarkable phase we're in right now with this translation of cell-based therapies, I think in large part due to this patient-specific nature that you pioneered. And a big part of that has been the integration of industry partners, right? That can assist with optimizing, standardizing, scaling these innovations that are coming out of research labs. And some of the biggest names in academia, Aviv Regev, Doug Melton, Sonia Schrepfer, many have taken leadership positions at big name companies to help foster the translation of their science. And your move to Roche was presumably rooted in a similar motivation. So maybe tell us a little bit about what drove that choice, how that experience has influenced you as a scientist, and where you expect that partnership to take those innovations that you have been piling up.
Yeah, well, so I was asked to be in the board of Roche, and Roche is essentially called Genentech in the US, right? So Aviv is a very close colleague. She basically held the same position as I did for Europe and China. She holds that position for Genentech in San Francisco. So it's basically anyway. So I was in the supervisory board as one of two scientists. Typically, Hoffmann Roche is a family-owned company, so typically there are two family members. There's a number of heavyweight business people from other industries, the airway industry or energy industry or food industry like Nestle or Lufthansa. And the two scientists basically have to keep an eye on the science of a pharma company. And of course, pharma is extremely R&D dependent. With drugs, your portfolio once a drug is registered, is approved by the FDA in the US or by the EMA in Europe, it probably still has another 10 years to go under the patent. And so every 10 years, you lose your entire portfolio and you have to replace it with new drugs that are even better than the ones you already had developed. So that's a huge challenge. And that's the reason why why science is so important in the pharma industry. So I must say I got intrigued when I was so the board essentially meets 6, 7, 8 times per year, a few days, go through sort of high level through what the company is doing, strategy, finance. I was in the search committee for being an executive of leading the R&D in Europe and China. And then sort of the search committee sort of turned on me and basically decided that I would be a better candidate than anybody on the list. So and then my wife just retired. We were in Switzerland. Headquarters of the company are in Basel. We've been skiing there for the past 25 years, share a family house there with a few brothers of mine. And then my family essentially turned on me because my wife retired from being a bone marrow transplant hematologist, and my two kids and their girlfriends loved the idea of us going to Switzerland for a few years, and they could cycle and ski and things like that. So eventually I agreed that I would do this for a limited number of years. I actually started in the week that I turned 65, which is in Switzerland the retirement age. So and I committed for a limited number of years. That is past now, so I stayed longer. But eventually I decided to, because combining it with my lab in Utrecht, I was a little bit joking about how they were, but I mean 40 people, PhDs, postdocs, technicians, it's a sizable lab. They publish, the papers need to be written, the grants need to be written, et cetera, et cetera. So I just stepped down. But the reason was essentially curiosity. So, because coming from academia, having never worked in a company, how is it, and then particularly a multinational company, how is that? Can I do it? I learned actually that if you think away the scale of something like a large pharma company, the challenges are very similar. It's basically you need a good team, right skills in the team. The people need to be able to get along. You need a strategy. but also you need to be flexible enough to, if an opportunity arises that's not on strategy, that you can decide to still go for that. Yeah, and then when I started, I realized I knew very little about drug development. I had a very, very good team that helped me. And we can discuss if you want a little bit more, how different it is to work to develop a medicine from writing a nice paper in a nice journal. But it is very different. And 90% of the project, the trajectory for projects had never been in my own scope. We discover targets and mechanisms, but the next steps, molecules, pharmacology, clinical trials, I'd seen from a distance. I had some idea about this, but I really had to learn this. And it's intimidating how complex it is and how many different scientific disciplines you need to develop a drug. And the statistics, probably 1 in 100. So if you have 100 projects, maybe one makes it. The cost goes up exponentially with every next step. So as in an academic lab, it's extremely important. It's more important to stop projects than to start projects. Because when you will not stop them, you'll go into the next phase. The next phase will be threefold more expensive than the phase you were in. You really have to think about the chances that is the one in 100 that will make it. And if the chance is not good enough, then it has to be stopped. Of course, sometimes you stop things that could have worked, but like in academia, but you'd never know.
Yeah, I mean, a lot of interesting parallels with what we deal with in academic labs. Like,
Yeah.
I'm just starting my own lab. And yeah, that's exactly right. I think more important than starting projects is learning which projects to stop and to understand kind of the financial ramifications of that as well. And, at an industry, at a company that's magnified, of course. So these are really important decisions to make. I mean, but nothing wrong with taking a new challenge kind of in the shadow of the Swiss Alps, right? So nothing wrong with that. So I wanted to actually take a step back and talk a little bit more about some of those initial discoveries, right? When your team first described LGR5 as a marker for adult intestinal stem cells, went on to generate these first organoids, intestinal organoids. I imagine you didn't always you didn't envision all the applications, the downstream applications that came many years later. And you talked about a number of them already, including these most recently snake gut and lung organoids, which is really cool. But looking back, what do you think was the most surprising or unanticipated use of these organoids? I mean, we always talk about the disease modeling, the drug discovery, all these kind of things, even microbiome interactions and virus applications like you talked about. What do you think was the most interesting or unique application that came out of that initial work?
Yeah, so let me think here. So if I bring in my drug development expertise now, so when we first saw these things growing, first of all, there were many unexpected observations then that they just kept on growing, that they stay normal, that they build a mini copy. We still trying to find out how exactly to do this, but they actually build a nice, fairly faithful copy of the tissue they come from. I thought this was going to be cell therapy replacing organ transplant. So what I've seen in pharma is how complex cell therapies are. And we've discussed this actually with the ISSCR quite a lot because, of course, the ISSCR has always had ambitions, and all the members of the ISSCR of developing stem cell based therapies. I now understand the challenges that pharma companies or biotech companies have to develop it. It's who owns the tissue. The logistics are totally different from drugs, antibodies or small molecules. If you think about where we use human cells, live human cells or organs, it's usually in the public domain, at least in Europe, like liver transplants, kidney transplants, heart transplants, blood banks. All are invariably public entities. And when they become commercial, there's a lot of challenges. There's the ethics as well. What I had fully underestimated, and of course the disease modeling, which is what in academia do a lot, trying to find the genes that play a key role in a particular disease. Maybe they're targets for drugs, but the pharmacology. I always thought of pharmacology as a pretty, sort of random science. You make a random molecule, you throw it at a tissue, something will happen. But and then that's your pharmacology study. So I thought, how interesting is this? There's no deep biology. now I realize that actually in this whole drug development process, probably the most unpredictable part and the key part is here I have, I have my target, I have an assay, I do a high throughput screen, I improve the compound. I can all understand that. Now I have a compound. Is it a drug? That becomes, so that's where currently all the animals are used, all sorts of, tests. Some of these tests are 50 years old, like the Ames test, bacteria exposed to substances that maybe are mutagenic. So I think that's where really the low-hanging fruit is, to build really reliable, and they have to be validated, have to be robust, they have to be standardized, they have to probably be miniaturized, so that the average technician in a pharma company can use these in a reliable way. But that's probably where most benefits currently could be gained in the next few years, particularly given the change situation in the US now, where the FDA wants companies to move away from animals as much as possible towards NAMs, and organoids would be in that group. So I mean, that was really maybe not surprised, but really an eye opener that actually that is a huge challenge in drug development, and that is probably where organoids have a really their natural habitat. Because you can make a kidney organoid, a liver organoid, a gut organoid, uptake of drugs, metabolism of drugs, toxicity of drugs, blood-brain barrier organoids exist, all these sorts of things, but they need to be validated and they need to be made more robust. I think that is what this field currently really needs. Cell therapies is still tough. I mean, we might have spoken, I don't know, we probably spoke about this 4 or 5 years ago or 3 years ago in our previous podcast. There's now actually a trial ongoing with salivary gland organoids in Rob Koppes in the north of Holland. He gets his first readout, so we'll be very curious to see. But that actually I had anticipated, because the organoids are from 2009, 2010, that maybe 5 years later we would have our first successes. We did have one success, that was cystic fibrosis personalized medicine, that has really worked well with the Vertex drug to identify patients that have mutations in the cystic fibrosis gene, but not the one that the drug was developed for. And their organoids turn out to be extremely predictive, and it have been used, at least in my country and some other European countries, for many patients. And then if the organoid test was positive, they would receive the drug and get it reimbursed. Other than that, it has been slow. And I've learned that basically, because cell therapy, I mean, it's essentially developing a therapy, a drug. This is a live drug. It's just extremely complex. Safety, production considerations, which patients, how do you organize the logistics, how do you produce, is much harder than I had envisioned when I was, but prior to my company experience.
Yes, for sure. I mean, the cell therapy in particular, the globally, I would say, universally, it's a slow roll, right? But only you would be, throwing up your hands at that hit rate. I think that's pretty good. You got one patient specificity element, and you got one cells in person. So, like, hey, kudos, because I think a lot of people would call it a day with that. Although maybe it's the hit rate that you're a bit disappointed with, because you do have all these innovations in your bag. You're probably cringing when you hear this every time, but you're known as the father of organoids, at least according to your Wikipedia page.
By now, the grandfather, I'm afraid.
Oh boy, no, you're still a young man. Even though you're retired, you're still pumping them out. And you yourself have acknowledged, I've looked in the literature, that Yoshiki Sasai, working in parallel to you and your collaborators, pioneered the development of like iPSC-derived organoid, rest in peace. But there's no question that you've earned the title, and the volume of organoid types that have come out of your group is extensive. And I like that you credit the lab, and even in absentia, they're still pumping it out. So clearly, it's the approach which you really encapsulated there in your narrative synthesis at the start. It's a model. You take the tissue, you make the connection with the primary, you get a specialist.
Wow.
Still,
well, there's a few rules in the lab, and then this has been highly controversial. So one rule is that if we start looking at something new, we don't come up with a hypothesis. So hypotheses are forbidden because hypotheses basically close your mind down, right? And because you're not going to nobody argues against their own beliefs, right? They always argue in favor of your beliefs. So the rule really is, we look at something new, we just kick this, we build a system that we think we can measure in, that is robust, and then we just kick it left and right, and we change things, and we knock out things, and we challenge it, and we infect it, or whatever it is, and we look and we observe. And then slowly, maybe you start seeing a consistent pattern. It has to be really robust. So if in an organoid it's not black and white, it's not going to be important. And so that's a good thing. So organoids are always, we keep them as simple as possible. So no fiber, no stroma. People always ask, well, where's the stroma? Where's the blood vessels? I think you lose the power of organoids when you start adding these, because every little layer that you add multiplies the controls you have to do, multiplies or basically dilutes out, makes the measurements very noisy. So basically, if you try to come up with a very simple system, you just kick it. A rule, some pattern appears. Maybe we allow a little bit of a hypothesis. It's immediately tested. If it doesn't fit exactly with what we see, the hypothesis is gone. We're not going to modify this, because the other thing, humans tend to make then almost there. It's a little bit more complex than I thought. And this will happen in multiple steps. And by the end, you have a hugely complex hypothesis that explains everything and nothing. So no hypothesis. That is one thing. The second thing is that don't do experiments where you already know the answer. That's how you train in university, right? You get a protocol. If you do everything right, you know what the outcome should be. But if you do experiments where you already know the outcome, you might as well do an experiment where you don't know the outcome, because that is science, right? So, but there's a very strong tendency, I've had it myself, I see it in the lab all the time, said, why are you doing this experiment? Yeah, because I got nice figure 1, B, C. He said, you already know what you're going to get, so why would you do it? And then the third one is, if you do an experiment and you don't know the answer, but if it's yes or no, it doesn't matter for any further steps. There's also no reason to the experiment. Because basically you spend a lot of time and then you get your yes or your no, and then the project stops. And so these are the sort of the simple rules that, and sometimes I make mistakes myself that I come up with. I believe very strongly that I know the answer to a scientific question. So that's a hypothesis. And then people in the lab tell me, Hans, no hypotheses in the lab.
It's always such a great conversation with you, Hans, because you say things that other scientists don't, because you're not supposed to say them. I remember you ended the last interview by saying, no one ever uses the grant money for what they said they were going to do.
Yeah.
And this is the same idea, is that, don't start with a hypothesis. I love that about your approach, mostly because you see the success that it's generated. So I think our listeners as well really appreciate that there's a lot of different ways to do science, and this maverick approach, I'd say, that defies convention, can be very successful.
Yeah, and it's about building tools that are simple and useful, like you're talking about, and then kind of deciding the most important questions after the fact, in a way. But you did allude to that, people are still trying to make these things more complex. Like we talk about vascularization, we talk about immune components, and incorporating these things into the organoids all the time. And I think in some applications they've been really important and successful. But I like how you alluded to the importance of the simplicity of the tool. But in spite of that, like in generalizing, what would you say is the most important next step for these organoids if you had to make them better, more complex?
So I think, for academic approaches, finding new mechanisms, finding new targets, et cetera, I think a next step would be combining. I mean, our organoids are always epithelial, right? They're not the IPS organoids. We cannot make brain. Well, we can grow fetal brain, but we cannot make retina, but we can make all epithelia, I think, by now. And epithelia, essentially, if you think about your internal organs, all organ-specific functions are executed by the epithelial compartments of that organ. You have to think about it, but like lung uptake of CO2 and O2 is done by the epithelial cells. pancreas, secretion of insulin, also exocrine, in the gut, in the liver. It is the epithelial cells that do and then the infrastructure of the organ is stroma, muscle, tendon, maybe nerves, immune cells, blood vessels. They more provide a more generic function, if that doesn't sound too disrespectful. So, but I think you can capture the function of an organ, the basic function in the simplest form in its epithelial cells. The next step would now be, so we had, for instance, one paper where we had this mutagenic bacterium, which we showed in colon actually creates a very particular kind of mutations that you then find back in colon cancer patients, but not in other cancer patients. So this is a very simplified microbiome, basically one species and its mutant, and then a very simplified human compartment, basically one epithelial organoid. And so I think that would be the next step. Do this with immune cells, do it maybe with fibroblasts, do it with blood vessels, but basically just combine two elements and find the rules of engagement between those two elements. And what we've always done, like for this bacterium paper, that actually essentially I had no knowledge about what's happening in my lab and Ruben van Boxer's lab, but basically the study was done when they showed it to me. So the prediction would be if this bacterium, it's an E. coli, a particular E. coli, if it indeed induces these kinds of mutations, we should find it back. So then we jump from a very simple human colon organoid and one bacterial species and its mutant that didn't cause these mutations, straight to a data set of 5,000 cancer whole genomes. And then we could see this particular mutation only occurs in colon cancer. And that is an association, of course. But rather than building an ever complex system in a lab, we could take that observation and apply it to some real world data. I think that is where I would see, but it's a style of research, that's where I would see the next step. So rather than immediately creating, there's labs that put 3-4 different organ types on a chip, right, multiple organs. The variability that you don't know how to control then is immediately beyond, well, at least my comprehension. So it would not be our style, but depending on what you invest. But just, I take one type of T cell, one type of macrophage in a context of one type of organoid and find the rules of engagement, I think, and do this really deeply. I think that is the next step. For application in industry, I would say, I already mentioned this, but standardize, make it more robust, automate, really important, microfluidics, whatever you need, automate it in simple machines, simple instruments. It's not, what I understand, not very hard. It's not my specialism, but that would be really needed. And then validate that actually what these assays produce is more predictive and more reliable than what currently the current, particularly animal-based models currently produce.
All right, I'm going to shift gears a bit here in terms of, we've been talking a lot about the practicality of these approaches and how to optimize and moving forward. But I want to take you back a bit because let's be frank. I mean, you made it. You're at the top. You got your seminal contribution. You got high volume, diverse research output, even in absentia. And you're moving this, although your hit rate you're dissatisfied with your hit rate, Hans, but I hate to break it to you, you're still moving research innovations into clinical practice. So you made it. And I think it's great what we're talking about here about, what drives innovation, curiosity, asking questions, where you don't already know the answer. And the rules, as you put them. I love that you have these rules, and it must be glorious to be a scientist, trainee, whatever level in your lab, because you kind of like have codified that freedom. But again, let's be frank, many at least, trainees in particular, I think are living within a system where it's kind of like, competition is ever thicker because there's so much less to go around and there's all kinds of screws that are turning. And so some people are it's hard to be so courageous. You've emphasized courage in your questioning and curiosity. And sometimes you feel like maybe you got to do the figure one where you already know the answer for the grant, especially as a trainee. So take yourself back to before you really had all this momentum, and trying some guidance for trainees in that atmosphere, how you can, and this is a tough question,
Mhm.
but how can you guide or balance that kind of staying on the path that is expected, but also, making those big
Uh,
swings?
uh, Yeah, but yeah, you're right that once a lab has made a number of discoveries, things get a bit easier. You get really good people in the lab that bring experience, bring their own ideas. Funding is, when the lab is larger, you can do, as I said, you can use your grant money for things that were not in the grants. You can always do enough that the grantees are happy about what happens with their funding. But yeah, if I go back, what is it, 35, 40 years when I started my own lab, with just me and one technician and a PhD student. That was sort of rather existential as it felt. And I guess everybody recognizes who's gone through that phase. And Owen, you're going through that phase apparently. Yeah, so you have to be a bit you have to be a bit lucky with picking the right people. So that is really important. The first few people are really important. I made the mistake, and I see many young PIs making the mistake that they don't realize that they are the world experts on what they want to do because they probably took this from their postdoc or have been thinking about it. And anybody who works for them is less experienced and has probably lots of other things going on in their lives. And so, but because it is so important that these people yield some success, I was probably too close to them, and I knew before they knew their outcomes of their experience, or I would know at 3 p.m. this afternoon they will develop their Western blot, and I'll be at the machine and see what it looks like and help interpret. And so I must have made them quite nervous. And I've actually tell the young PIs here in my institute that, provide some freedom for people to make mistakes, because sometimes they are not mistakes, they're basically they create a new insight, and also people need to be able to have enough freedom around them. But so I think another thing is, you mentioned the word courageous. Be courageous and don't go where everybody is. So there's always these hypes, and I've seen a large number of these, and everybody jumps at them. And for instance, now if I would start a lab, I don't know what you're going to be doing, but the microbiome, for instance, is in the midst of a huge hype. If you join that hype, it is going to be really tough because to do something original, or even if you find something, the final answer to an important question, there are probably already 20 answers in the literature. How would anybody know that the answer number 21, your answer, is the only good one? And it's hard to publish. So go into areas like snake lung, where there's nobody else, but where you can probably find interesting things. And that people find that hard because you have very little ground under your feet, as we say in Dutch, because you don't really know if you can do anything significant. So you have to have trust in yourself and courageous. That we've done it multiple times. Just move into an area like the gut stem cells. I started all this work was done in the 60s, 50s, 60s, a little bit in the 80s. There's essentially almost nobody studying normal gut. Everybody was studying celiac disease, IBD, colon cancer, but not the healthy gut. And I realized, well, these are probably the most interesting, at least I thought, the most interesting stem cells in the human body. Nobody's trying to find them. And so we moved there, we found it. It took about 10 years for the clinical people who were driving that field to realize that this has happened. I was mostly, in the basic science, and then slowly, and particularly when organoid technology became feasible and companies started selling the reagents, made it a bit easier. Yeah, but so don't shy away from going into an area where you know very little and where there's nobody else.
Mm.
Because if you find something, it's definitely going to be original, and there's no competition, and it has to be interesting. That's the one thing. You have to be able to write the story. That's the other thing. Story writing is really important. So a paper is essentially uh, an ordered set of random data. You order it in a particular way to tell your story, and that is I learned over the years. Your title, your abstract, are really important.
Mm.
And I spent lots of time on this, like a haiku title. Basically, get the keywords in, raise interest, some curiosity. Same thing, the abstract. But I like the sort of the nature or the cell type. These very short, where every word has to be the right word. It's like a poem.
Mm.
And without a rhyme, though.
Yeah.
Usually.
No, that's very important, the importance of telling a story, and also to make a stem cell organoid relevant analogy, the importance of finding your niche, right?
Yeah, yeah,
Um,
yeah, yeah.
yeah. And yeah, I mean, like my example, I could be another IPS cardiac disease modeling biologist and do what Joe Wu does, who is my graduate mentor and has been amazing. Or I could find my own niche. And as the listeners of the show know, my niche is sending things to space, actually, using low gravity to see what happens to organoid biology. And it's a little different, but it's allowed me to kind of establish myself as a leader in that subfield. So it is definitely important. But also important is the ecosystem in which you're growing, in which you're developing as a scientist. And I mean, you've worked at the intersection of basic stem cell biology, translational applications, and this European ecosystem. So you've gone back and forth industry, Hubrecht Institute, and just more broadly have experienced this European basic research infrastructure. So tell us, what do you think are the unique strengths or advantages of that particular ecosystem? And how do you see that as important for translating these organoid discoveries?
Yeah. Well, so Europe is of course not a single country.
Sure, of
You
course.
mean the scientific environment in different European countries is very, very different. And is in part it's the funding system, it's the way that the university hierarchies are set up, but it's also national cultures. So what I saw, I did my postdoc at Harvard, and I believe that the only place where you can do good science is Harvard, because that's what I saw. People work, then I don't know how it is now, but we used to work like 80 hours a week. Christmas Eve I was in the lab, or Sunday evenings I was in the lab, and not the most collegial atmosphere, if I can say that on this podcast. So everybody was out on their own, and if I make it a little bit, so I decided I love science. I had a great time in my postdoc, but I also decided I want to be friends with my colleagues. I don't want to be competitors with my colleagues. And in my lab, people really need to be happy in my lab. They need to come to my lab because they want to be there every day of the week. So in my lab, actually, I don't think people, I wouldn't know, but probably don't work more than an average working week. They're very flexible. That's what I tell them. Come in the first time you can do something to your experiment, please come in and do it. Because if you postpone it because your aunt is visiting over the weekend, you lose 2 or 3 days of your PhD or your postdoc. But so what is really a strong cultural element in Holland is that so in Holland you can be ambitious, but you always have to be part of a group. So you do things together. You do it in a lab. People team up, help each other. So when somebody publishes a really nice paper, everybody else comes in and celebrates. But also when somebody has something that doesn't go so well, the lab is also there to basically stand around them and help them. And then in collaborations, I always emphasize we are fully open. We are transparent. We share everything we have, our reagents, our insights. We share observations that we haven't published yet. Almost always this is positive because people appreciate this and then reciprocate this, maybe in the moment or maybe 5 years later. And there's very little abuse of this, and there's a lot of distrust in academic science. So the culture is very important. So Holland brings this totally non-hierarchical culture. So I mean, people call me by my first name, and they disagree with me. And so you would not see that in some other, I will not name country, but in some other European countries where the professor comes in and when you do what the professor says. And you can think of some other countries in the world where this would be the case. That's not good for science. It's great in an army, but not in science. When you want to have create like the E. coli, the cancer-inducing E. coli, that was done entirely by the PhD students. They realized we have the freedom to do this. We should inform him at some point, but we can basically see if this works and then show it. So that's part of the ecosystem. So I think the UK has quite a bit of this. I think the Scandinavian countries are really good in this. I see it in Switzerland, well, in Germany. Other countries have less of that. And so that is a strength. There's a big difference between the way the universities in the US are set up and the universities in Europe are set up with lots of tenure jobs and Americans would look at the European system and say, well, everybody gets lazy, they have a 10-year job, why would they still work? We would look at America and you're never sure about your retirement or whether you have a job the next Monday. So both systems have I guess, pros and cons. There is something, when I moved back, I thought, why would Dutch science work? Because, you get your 10-year position with 35, there's no way they can fire you anymore. So why would it work? I mean, there's also an important element in this existential threat that you lose your job if your next experiment fails, or if you don't get your next grant, or you don't get your paper in Nature. There's much less of that in Europe than there is in the US. For some people, this takes the urgency out of everything they do. For other people, it just creates a lot of space in which they can be creative. So there's pros and cons to both systems, I think. And I cannot deny that American science has been extremely successful in the past 3 or 4 decades.
Yes, I would agree with that, but you wonder about how happy the scientists are. And what you're describing really sounds idyllic in that way that, I would think that you're coming to work, you're going to the lab because you love it, but I'm projecting. But I would think that, like you said, picking good people, you surround yourself with people who have joy in the work, and that sustains you beyond tenure or whatever the plaudits, accolades maybe. So I'm voting for your model, Hans, and you've been very generous in sharing, frankly, what that model is, and I think really are a good example of how it can be successful. I mean, just in the last 5 years, I was looking at, just a small snippet of some of the output, which you attribute to the lab members, rightly, but I think you had a lot of leadership role in, and it's like all kinds of organoids, lingual, the taste bud, salivary gland. We covered that one. That was amazing. The thymic organoids, airway, submucosal gland, respiratory, conjunctiva, lacrimal. Remember the organoids that cried? I had me weeping, Hans. And that's not even to mention all the cancer organoids, and like you said, the getting into the animal, the canine pituitary. You mentioned the snake gut. I didn't know about that one. I saw snake venom gland. I mean, you're fully in mad scientist territory right now, Hans, and I'm sure that the lab is happy to welcome you back. We really appreciate you taking the time on your son's birthday, no less. A shout out to him. Happy whatever year. You got a great pop, the father of organoids, and your dad too. Anyway, Hans, this has been so much fun, and we got to have you back in another 5 years and probably another 100 organoids later. Really appreciate your time.
Okay. Very nice talking to you guys.
All right.
Have a good day.
All right, y'all. That brings us to the end of this episode. Fireside chat with the father of organoids. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. If you enjoyed this episode, please feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELLpodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests. Not going to do much better than the one we had on this episode, but we're going to try. We'll be back in your ears in a couple weeks. Thank you guys so much for listening.